Hi everyone. So let's go ahead and finish up this monster section right here, which is section 5. This is still going over the pulmonary ventilation. So remember in the last video we talked about the mechanics of breathing. We talked about how it relies on this pressure gradient. But in order to get that pressure gradient, you had to change the pressure in the thoracic cavity. In order to change the pressure in the thoracic cavity, we have to change the volume of the thoracic cavity. In order to do that, it's going to rely on these muscles contracting and relaxing. So the thing that's going to make those muscles contract is going to be, of course, the nervous system. So one disclaimer I need to talk about before we go through the whole nervous control of breathing is the fact that the nervous control of breathing and really the, I guess the exact structures and what their functions are is still very, very new. You'll read some books that will go in and talk about, hey, this is the center, this is the group that does this. Like this book. This book will talk about how the ventral respiratory group is going to be the main group of nuclei that is going to cause inhalation and exhalation. But you read another book and it talks about how it's the dorsal respiratory group that's going to do it. So there's still a lot that's not known about how exactly the nervous system is going to control breathing and what structures are going to control it. They're pretty sure on the structures. They're pretty solid on this VRG and the DRG, but they're just not 100% sure if this is actually what it does. We're going to go with what our book says. Just make sure you know that if you do go to an external website or another video, they might be saying something completely different, but you're going to be tested on what your book says and what we talk about in this video. So with that being said, let's go ahead and talk about it. In the nervous system, the respiratory center is going to sit in the brainstem. You're going to have a couple of groups that are going to be found in the medullary respiratory center, and it's going to be the ventral respiratory group, and you also have the dorsal respiratory group. They're named that from, let's see, where did it go? Where's the picture? Here it is. So look at that picture. You are going to have, they're going to be named by really just where they are in the brainstem. You're going to have that dorsal respiratory group right here, and then there's the ventral respiratory group right there. And then this over here is going to be the one found in the pontine respiratory group. So those are going to be the main places, the autonomic nuclei that's found within the brain that's going to help control and coordinate breathing. So let's go ahead and take a look. With the ventral respiratory group, the VRG, it contains both inspiratory and expiratory neurons. So we'll talk about how you're going to have this stimulus that's going to activate the inspiratory neurons, and then that signal is going to pass the axons from the upper motor neurons extending from the VRG is going to go to the spinal cord, and they are going to synapse with the lower motor neurons. And then those are going to project to the respiratory muscles. They're going to project to the ones that we are going to, oops, I need to go in and put my pen on, sorry about that. You know, the ones that we're going to concentrate on, where am I? Right here, is going to be the diaphragm and the intercostals, and that's it. Because again, the group that controls the accessory muscles is still, there's a lot of controversy there as well. So that's the ones that we are going to look at, is just the diaphragm and the intercostals. So look, they travel down, the ones that innervate the diaphragm travels down the phrenic nerve, and the ones that innervate the intercostal muscles travel down the intercostal nerves. So you might remember that phrenic nerve from 2401. You also have the DRG, and the DRG is going to initiate neural impulses for inspiration and expiration. So we're going to talk about how there's a lot of stimuli that comes into the dorsal respiratory group, and then the dorsal respiratory group tells the ventral respiratory group what to do. So it kind of like, hey, we need to breathe a little bit more. The ventral respiratory group will be, okay, I got it, and then it will send the signal down to the diaphragm and the intercostal muscles to increase the breathing rate that way. So you also have the pontine respiratory center, and this is mainly there to facilitate smooth transitions between inspiration and expiration. So it communicates with these respiratory centers in the medulla. It goes in and kind of controls how big of a breath we take or how much we take in as far as inhalation. Damage to it causes erratic breathing, like it can cause you to gasp for air and then have erratic exhalations. So that's one thing that they do know is what happens when the pons is definitely is damaged, but they're still not 100% sure as to the pontine respiratory group's main function. So some books will even talk about pneumotaxic center and the apneustic center as being part of the pontine respiratory group. So again, you might see those on another website. This is what's happening if we just kind of look at it in general. The book goes through it pretty extensively. We're just going to go through it very briefly. Ventral respiratory group gets a signal that it needs to initiate inhalation. So those inspiratory neurons are going to be activated. We're going to get that signal. Remember, upper motor neuron, lower motor neuron is going to go in and cause the muscles to contract, the ones that we need. And then that is going to cause the normal quiet inhalation. And then it's going to say, hey, we need to exhale. So it's going to inhibit any kind of activity. So it's going to become inactive. That's going to allow those muscles to relax, and that is going to allow that quiet exhalation. So this should be, you have about 2 seconds of this stimulation that's occurring to cause inhalation, and then exhalation, this inhibition, is about 3 seconds. So if you kind of put that together, this normal respiratory cycle, which is a breath in and a breath out, normal respiratory rate or respiration rate is going to be about 12 to 15 seconds. Sorry, 12 to 15 breaths per minute. So that's where it comes from, is this about 2 to 3 seconds to inhalation, 3 exhalation. So that's the gist of it. Let's talk about how the DRG is going to get involved a little bit. Really, we don't even talk about the pontine anymore, the pontine respiratory group. I don't care about that one. So we all know that our breathing isn't that rhythmic. It's not 2 seconds out, or 2 seconds in, 3 seconds out, 2 seconds in, 3 seconds out. We, of course, are going to need, any time that we exercise, it's going to change. Anytime that we have issues with chemical levels, anything like that. Anytime that we are going in and singing or talking while we're trying to breathe is going to change as well. So we're going to take a look at how breathing rate and depth is going to be changed with reflexes. And these reflexes are going to respond from sensory input from receptors. Chemoreceptors, we'll talk about 2 chemoreceptors. They are going in and constantly monitoring chemicals in the blood or in the cerebral spinal fluid. Those proprioceptors are going to be found in the joints, muscles, stretch receptor, these nice baroreceptors we're going to have. And then also we have some that are picking up irritants as well, irritant receptors. So what happens is all of these receptors right here are going to talk to the DRG and tell it, hey, a little bit too much hydrogen ions, or hey, there's a lot of activity going on, or oh, we're getting a little bit too much stretch. So they go and talk to the DRG, and then the DRG goes and talks to the VRG. So they cannot talk to the VRG. They are not allowed to talk to the VRG at all. So again, the VRG is going to go, okay, great, I'll go in and increase my breathing rate. I will go in and instead of 2 seconds and 3 seconds, it'll be, 1 and 2, or, it's going to increase the inspiration and expiration that way. Or it can go in and change the depth by going in and stimulating those accessory muscles. So if you need to breathe deeper, you're going to get that forced inhalation and forced exhalation involved. So let's first take a look at these chemoreceptors. You have 2 internal chemoreceptors. You have central chemoreceptors, and then you have peripheral chemoreceptors, and they are both very important. Probably one of the most important factors that we have at regulating changes in breathing. What they're going and doing is they're constantly monitoring hydrogen ions and respiratory gases, either in the blood. These are responsible, the peripheral chemoreceptors are responsible for blood, or in the central, the cerebrospinal fluid is going to be central chemoreceptors. So just remember, that starts with a C, and so does that. That's how you can remember cerebrospinal fluid, central chemoreceptors. And of course, remember that's found in the brain. So what they're going and doing is constantly monitoring pH changes in the cerebrospinal fluid, and it's because of the blood PCO2. If you don't like this P in front of it because we haven't talked about it yet, then ignore it and ignore all the P's right now in front of CO2 or O2. It has to do with partial pressures that we haven't mentioned yet. We'll get to that later on. But let's take a look at this picture. Look, so remember that whole blood-brain barrier and how that is going to work. So you do have carbon dioxide that can diffuse over to the cerebrospinal fluid, and then what happens is that it combines with water, and then as soon as it combines with water, it is going to dissociate into a bicarb and a hydrogen ion. Now the issue we have with this is the fact that the cerebrospinal fluid doesn't have any buffers to take care of that hydrogen ion, so it accumulates in the cerebrospinal fluid, which is not good. So the way to get rid of it, get rid of this hydrogen ion, or make sure that it doesn't collect anymore, is to get rid of this blood CO2. How do you get rid of your CO2? You exhale, don't you? Yes, that is exactly how you get rid of it. It's in the bloodstream, so we need to exhale it. We need to get it to the lungs, and we need to get it out as quickly as possible. So that's what's going to happen, is that that's going to be relayed. Let's go back to this. Look, it's going in, and this is what's really cool, is these are the central chemoreceptors. They lie right next to the VRG and DRG. So they tell that DRG, hey, we've got too many hydrogen ions in the cerebrospinal fluid. And the DRG is like, okay, well, then I'm going to increase. So I'm going to put a positive to show that it's going to go in and increase the respiratory rate. So that's how it's going to do it, so we can get rid of that carbon dioxide. Because look, and this is what's happening in the blood, so it's going to be a very similar process. Look, because the more CO2 you have, the more hydrogen ions you are going to have, and that is going to change pH. If you can't remember pH and hydrogen ions, then you need to go back to your 2401 notes, or back in the chapter in 2401 that talks about it, so you can remember it, because it's going to be important here, it's going to be important in urinary, it's going to be important in fluid and electrolytes. So I got to make sure that you understand that relationship between hydrogen ions and pH. Let's take a look and see what happens with these peripheral chemoreceptors. They are the ones that are constantly monitoring respiratory gases, but also pH as well. So look, they're going to be the same place. Look, that the from the cardiovascular system, right here in the aortic arch. So you have aortic bodies, and also in the carotid. So the carotid bodies. And what they are doing is they are going to be stimulated by changes in hydrogen ions or CO2 and oxygen levels. So, but we're going to put a little star on this. We're going to talk about it here in a second. It's mainly going to be stimulated carbon dioxide, but also look, it responds to hydrogen levels produced independently of carbon dioxide. Not because of this, but because of some other thing that's causing a buildup of hydrogen ions. Maybe a person, the book talks about how if an individual has kidney failure, then that is going to change their hydrogen ion concentration. If a diabetic is in ketoacidosis, that goes in and causes a change in hydrogen ion levels. Lactic acidosis is going to cause a change in hydrogen ion levels. So anything like that is going to cause changes in hydrogen ion levels. So it talks about carotids are going to send it via glossopharyngeal nerve, and then the aortic chemoreceptors are going to change it via the vagus nerve. And so you should be able, you can see it right here, it talks about glossopharyngeal and vagus nerve there. But this is the most important thing to know right here. Blood CO2 is the most important stimulus affecting breathing. If I go back and look at this, I forgot to talk about this. So it can stimulate breathing if oxygen levels drop, but it has to have a dramatic drop in oxygen in order for these peripheral chemoreceptors to tell those medullary centers what's going on. Again, it's mainly this PCO2, the carbon dioxide that's going to be in the blood. Because look, raising the blood CO2, PCO2, by just 5 millimeters of mercury is going to double your breathing rate. But look, arterial oxygen must decrease quite a bit from 95 to 60 That is a lot. What is that, 25? Yeah, 25 millimeters of mercury to have any kind of major effect on breathing rate. And usually this doesn't happen unless altitude changes. So other than that, we'll talk about how even if you hold your breath, this is going to happen first. This accumulation of carbon dioxide by just 5 millimeters of mercury is going to happen first, and that's what's going to cause you to breathe. So we'll talk about more about this in hyperventilation and hypoventilation. So again, going back here, this is just talking about what I already mentioned there, how carbon dioxide combines with water and to form carbonic acid, which of course splits and shoots off that hydrogen ion. And then I talked about how there's no buffers in the CSF. Let's take a look at some other factors that are going to cause a change. So I went back, let's go back to this one here. I talked about how this is going to cause an increase in breathing rate. So look, an increase in CO2, increase in hydrogen ion levels, those are going to also cause an increase in breathing rate. We're going to talk about how this one causes an increase in breathing rate, but these two that we're about to talk about cause a decrease in breathing rate. So let's look at those. Proprioceptors are going to be those receptors that are found in the joints and muscles. So they are going to be stimulated by body movement. So anytime you start to walk faster, or any time that you start to jump up and down, anything where it's going in and stimulating these joints, saying, hey, this person's moving a little bit more, so maybe we need to increase the depth of our breathing. That's the main thing that it does with these proprioceptors, is it actually goes in and increases the depth instead of the rate. So we'll talk more about that concept when we talk about exercising later on. So baroreceptors and irritants are the two that are going to go in and decrease breathing rate and depth. So the baroreceptor is that stretch receptor. It's found inside the visceral pleura, and what it's doing is it's constant, and also look, it's the bronchioles as well, but it's constantly monitoring how big of an inhalation you are taking. It's making sure that your lungs don't inhale so much that they are going to rupture. So this is a nice little protective mechanism that we are going to have to make sure that the lungs don't overstretch. They are going to go in and shut off inspiration to protect overinflation. This is also called the Herring-Brewer reflex. So that's another term that you can use. But again, it's going to cause a decrease in the breathing rate and depth. Irritants. So these receptors, you have a bunch of irritant receptors found in your nasal cavity, down in your trachea, in your bronchi. So they are either going to cause you to sneeze, or they're going to cause you to cough. And as you can understand, it's going to change it because you're going to temporarily stop breathing so you can let out this explosive blast of exhalation. And that's what a sneeze and a cough is. So that is what's happening. It's going to be, of course, stimulated by particulate matter. So anything that maybe gets in your nose and makes you sneeze is trying to get rid of it. That's what it's doing. Or anything that gets in your lungs that your body's trying to get rid of that way. So if we go back and look at this picture, we can see how this one's coming from the cerebral cortex here. It doesn't show anything about the hypothalamus or the limbic system. But what's interesting about it is, look, that cerebral cortex, the output, look, it's like, nope, I don't need to talk to you, VRG and DRG. I'm going to go ahead and go straight on to these muscles and do my thing, my own thing. There's the accessory. So that's what it's looking at that way. But what's that doing is it's actually going in and changing up. So it's from the frontal lobe of the cerebral cortex. So it's going in and controlling voluntary changes. So anytime that you have some kind of voluntary activity you're doing, like me constantly talking right now, if you're singing, if you're doing the Valsalva maneuver, anything like that, it's going in and controlling and skipping the VRG completely and controlling those muscles that way. The hypothalamus will increase breathing rate if your body temperature is going to increase. It decreases breathing rate whenever your body is cold. Limbic system alters breathing rate whenever with emotion. So if you get angry or have anxiety or anything like that, usually your breathing rate is going to increase. So let's kind of, so that's basically it for the nervous control. What we're going to do is kind of put everything together and take a look at this whole, some of the factors that might cause issues with airflow. So remember, there's one thing that we've talked about so far, only one. We've talked about pressure gradient. We've talked about how important it is to have a pressure gradient, and that is what's going to really drive air going into and out of the lungs. And that's what airflow is. It's just air going into and out of the lungs with each breath. But now we need to talk about resistance. You guys know resistance. Resistance is going to be any factor that opposes flow. So in this case, we are looking at airflow. So we'll talk about how it's within the airways, the lungs, the chest wall, anything like that. This should be familiar to you guys. You should remember this nice little equation from the cardiovascular center. This F is referring to airflow. Here's that change in pressure. And obviously this is going to be the difference in pressure between mainly that intrapulmonary and the atmospheric pressure. But look, resistance is going to be in there as well. So remember that flow and pressure are directly related. Whenever you have a larger pressure gradient, you have a larger flow. That's what happened with forced inhalation. You had a bigger expansion of the volume, which caused a bigger pressure gradient, which allowed you to take in so much more air. And the opposite is going to happen. If you decrease pressure gradient, you're going to decrease your flow. And then it's going to be inversely related to resistance. So if you increase resistance, you decrease flow. So let's take a look at how this is going to change. So since I just talked about resistance, ways that resistance changes is because of the elasticity. Remember, lungs need to be very elastic. When they're not elastic anymore, that's going to cause resistance, and that's going to decrease your flow. Bronchial diameter, just like blood vessel diameter. Yes, we know how that works, right? And then collapse of alveoli because of surface tension. So those are going to be some factors that make it more difficult to move air through from the atmosphere to the alveoli. But some things that, let's talk a little bit about pressure gradient. Pressure gradient doesn't change a whole lot. It might change with altitude, right? Didn't we talked about that whole issue with altitude and how that's going to cause a change in the atmospheric pressure. So anytime you change atmospheric pressure, it can cause a change in the pressure gradient. We'll talk about altitude later on when we talk about gas exchange a little bit more. Let's look at some of these factors with resistance. That's what we're going to concentrate on more. So anytime that you have a decrease in the elasticity of the chest wall or lungs, that increases resistance. So in healthy young individuals, your lungs are very elastic, so you should have plenty of airflow. But if you have any issue with maybe any kind of skeletal issue that doesn't allow your chest cavity to open up like it needs to, or let's say that you have pulmonary fibrosis, that goes in and replaces scar tissue, it replaces the elastic connective tissue with scar tissue, so it's not elastic anymore. That's going to increase resistance as well. And just naturally, as we age, we lose some elasticity as well. So those are going to be some issues that increase resistance that are going to go in and decrease airflow. Bronchial diameter, that's going to vary inversely with resistance. So anytime that we decrease the diameter, we increase resistance. So a couple ways that we decrease diameter. One way is bronchoconstriction. So bronchoconstriction can occur from parasympathetic activation. Histamines, so if you have an allergy to like a peanut, histamines are released and it actually causes your bronchioles to constrict. And cold weather can do it as well. So not as much as like if you have a bad allergy to a peanut, but it still can happen. Other like bronchitis and asthma will go in, it can cause bronchoconstriction, but it can also go in and cause a bunch of mucus to be filling up that diameter, and that decreases the diameter even more. So that causes resistance, and people have issue with airflow with asthma and bronchitis. If you increase the diameter, like with bronchodilation, with sympathetic nervous system, that is going to decrease resistance, and airflow is fine. So epinephrine, whether it's through a pen, an EpiPen, or if your body's naturally releasing it, is going to cause that bronchodilation. And then collapsed alveoli is going to increase resistance. And this is mainly because of surface tension. And we talked about surface tension. We talked about the importance of surfactant. So this issue really doesn't come about unless it's with a premature infant. With premature infants, so what happens is surfactant is not produced until the fetus is about 7 months fetal age. So if you have a premature infant, they're not producing surfactant, so their alveoli are going to naturally collapse with every exhalation. So they tend to have to go in and take these huge inspirations, these huge breaths in to try and overcome that resistance, and a lot of times it doesn't work because they're just not capable of doing it. So they go into respiratory distress. So that's what that respiratory distress syndrome is. Sometimes you can see it as ARDS, acute respiratory distress, or acute respiratory disorder, but it's typically acute respiratory distress syndrome. So that happens more often with babies because surface tension and the lack of surfactant. Compliance is another thing that's going to go in and kind of change resistance because it's determined by surface tension and elasticity. Compliance has to do with the ease at which the lungs are able to expand because of the elasticity and because of the decrease in surface tension. So if it's very easy for lungs to expand, they have a greater compliance and a better airflow. So we'll talk about emphysema and how people with emphysema have issues with compliance because number one, it is going to go in and mess with this elasticity, but another component as well. So as you can see, a lot of these respiratory diseases that I talked about, any anatomical abnormalities are going to cause issues, mainly with increased resistance, but they can cause issues with pressure gradients, but it's more of them go in and cause issues with resistance. What happens with these respiratory diseases like asthma and bronchitis is those muscles of inspiration have to work so much harder because they're going in and trying to forcefully inhale so often. And so that causes a 4 to 6-fold increase in energy needed. So their total body expenditure going towards breathing goes from 5% up to 25%. So that's an enormous amount of your energy expenditure that's devoted to breathing. So individuals that have problems with asthma and have problems with bronchitis and things like that, they are going to be exhausted just from trying to breathe. So now what we're going to be taking a look at from here on out is just these different volumes and capacities. This is going to have a little bit more to do with your lab. The lab simulation you're going to do, part of it is going to be looking at these different volumes and capacities of your lungs. So before that, let's go ahead and, well, let me back up. We're not going to go through all of this in lecture, so you're not going to be responsible, well, you'll be responsible for some of it, whatever it is that I mentioned that you need to make sure you know for lab. So just please make sure you follow along carefully with what I send out about what you do need to know for lab and what you don't need to know from this lab, because it is quite a bit of information that you'll go through in that lab simulation, so you'll want to make sure you follow along very carefully. But pulmonary ventilation has two definitions. This is the one that we've been talking about so far. But another definition is the amount of air moved between the atmosphere and the alveoli in a minute. 1 minute right there. So in order to calculate pulmonary ventilation, you have to take tidal volume. I mentioned tidal volume in the earlier part of this section where I talked about anytime that you have that natural inhalation, just a quiet inhalation, that is going to be the amount that you take in is 500 milliliters. Can't write with this pen. I apologize for that. So there's 500 milliliters written out for me. And also, we have to look at respiration as well. Respiratory rate, you have 12 breaths per minute, and that gives us our pulmonary ventilation. That's the typical amount that we are breathing in, amount of air that we're breathing in 1 minute. Let's talk about the alveolar ventilation. Alveolar ventilation is going to be the amount of air that actually gets to the lungs. That's important because if you think about, yeah, we're taking in this 500 milliliters, but a lot of it, well, I don't want to say a lot, 150 of it is going to be stuck in the conducting zone. And remember, conducting zone, all it is just the air is hanging out. It's not, there's no gas exchange that's going to happen. So that's called the anatomic dead space. The air that's sitting in the larynx, in the trachea, in the bronchi, just sitting there waiting to reach the alveoli. So in order to find the actual amount of air that we can use for gas exchange, you have to take that tidal volume, which is 500. Let me see if I can write it out. Nope. And then you have to take the anatomic dead space, which is 150, and then again you multiply it by 12. And then you should get 4.2 liters, 4.24 liters, I think, per minute. Yeah, 4.2 liters per minute. So you have to go back, that's a milliliters, that's a milliliter, so you have to convert it. It comes out to be 4,200 milliliters. And then if we want to change that to liters, that's where we get 4.2 liters per minute. So remember that the pulmonary ventilation was 6 liters per minute, but what we can actually use for gas exchange is 4.2 liters per minute. It's not too bad, right? No, it's not too bad. We can actually increase that if we go in and breathe deeper. If we do a forceful inhalation, forceful exhalation. But if we do that, mainly that forceful inhalation, we can go in and increase this number. It's no longer called tidal volume. We're going in and adding some more air, and that's going to increase this number. So the spirometer is what you guys are going to be, one part of the lab that you're going to be looking at. If you've ever gone to the doctor, a pulmonologist, or even my son's allergist, he does a spirometry whenever he checks his asthma because of his allergies, then you know what a spirometer is. And what it does is it measures the volume of air that enters and leaves the lungs. So this is really good for diagnostics and assessment tools. There's 4 volumes that can be measured in spirometry. Look, we know what tidal volume is. IRV is that thing that I just talked about, how you want to take a really deep breath. That's going to be that amount of air taken into the lungs during a forceful inspiration. ERV is going to be the amount of air you can force out all the way, that forced expiration. And then you have a residual volume. It's going to be that little bit left over in the lungs that following a forced expiration. So all these can be measured in some way or another by a spirometer. There's other capacities that you can go in and find if you want to do some formulas too. You don't have to worry about these. Look, total lung capacity is going to be the total amount of air that the lungs, that can be in the lungs. Look, males and females, of course, are going to be different. So they are going to have a bigger lung capacity. I mean, that's why, you know, sometimes they can run longer than you, or sometimes they can't, right? It really just depends on the man and the woman. But they have such bigger lung capacities. So anyway, not much more to say about that. You can chart it on this little chart here. We're not going to be going through that. But there's additional respiratory measurements that doctors can use. And a lot of doctors will actually use this called forced expiratory volume, or an FEV. This is going to be the percent of vital capacity that can be expelled in a specific period of time, like 1 second. So what they make you do is they, again, if you've ever done this before, a spirometer before at the doctor's office, it's usually hooked up to a computer, and they make you blow on this little device, and the computer checks it. But they make you inhale as much as possible, and then they make you exhale it as fast as possible and as much as possible as you can. And they are going in and measuring to see, you know, how much can you actually exhale in that 1 second. And a healthy person, 75 to 85%. But if you're having issues with asthma or anything else at that point, it's going to be lower than that. Emphysema also is going to decrease it. So that's about it for this giant chapter, just talking about pulmonary ventilation, right? So the next section that we're going to be taking a look at is gas exchange. So until next time.